Hi, everyone. My name is Nader Morshed, and I'm excited to talk to you today about the project that I worked on in Forrest White and Li Wei Tsai's lab. And the goal of this project was to perform a signaling analysis of Alzheimer's disease in order to better understand which signaling networks were changing in the disease in order to better develop therapeutics against Alzheimer's disease. And to begin with a bit of background, Alzheimer's disease is the most common form of dementia that's characterized by neurodegeneration in regions of the brain that are associated with memory and cognition. And the brains of Alzheimer's patients have been characterized as having these two key pathologies: amyloid beta plaques, which are composed of short protein fragments that bind together into large aggregates, as well as neurofibrillary tangles, which are composed of a protein called tau, which is phosphorylated in disease. And this phosphorylated tau binds together into large aggregates within the neurons and contributes to their degeneration. However, there's a hidden player that's not visible in this image here, and that is the microglia, shown here in green. The microglia are the brain-resident immune cells that play an important role in keeping the brain free of foreign pathogens. But these microglia have a complex role in the brain and contribute to learning and memory by forming many tight junctions with the neuronal synapses, as well as the neuronal bodies. And they signal back and forth to these neurons in order to mediate learning in the brain. And in disease, microglia have an important role in breaking down protein aggregates. And the reason why we became interested in these cells was a result of a phosphoproteomics analysis that we performed for our project. Phosphoproteomics is a technique that allows us to take a brain tissue dissection, and in our case, we used brain tissue from several different mouse models of disease. And this technique allows us to enrich for the phosphorylated sites on proteins and then analyze the abundance of these phosphorylation sites using mass spectrometry. And what we found in our analysis was that there was this protein, SIGLAC-F, that had increased phosphorylation across all of the different mouse models and brain regions that we looked at. And to give a little bit of background on the SIGLACs, these are a family of immunomodulatory receptors that bind to sialic acid, which is a glycan, and alter the cellular signaling immune response of the cell that is expressing this receptor. And so SIGLACs have an important role in recognizing the sialic acid pattern present on cells and allows an immune cell to distinguish host cells from foreign pathogens. However, these SIGLACs could have a pathogenic role in disease. And so we hypothesized that this receptor was expressed in the microglia in the brain. And so that led us to take this image, which is up in the gallery today, showing the microglia in magenta expressing SIGLAC-F in cyan in close proximity to an amyloid beta plaque, shown in yellow. And you can see that the SIGLAC receptor forms a tight surface around the dense core of the plaque, suggesting that it may be altering the microglia's ability to respond to this protein aggregate. And so going forward, we'd like to better understand what is the functional role of SIGLACs on microglia in diseases of the brain. But I'll cut it short here and just thank the people who supported this work. The White Lab and Tsai Lab are full of a bunch of amazing and wonderful people, and especially Forrest White and Li Wei Tsai are great mentors who taught me a lot. And I'd especially like to thank Alexi Knott and William Ravenius, without whom this work would not have been possible. And I'd also like the institutes. I'd like to thank the institutes and funding sources who supported this work. And if you have any questions, just ask them in the chat. Thank you for your time.